Hi, welcome back. We are still in the central nervous system, now talking about the meninges, which if you've done the lab already, you are familiar with the meninges. They are three layers of connective tissue that cover and protect the central nervous system. They also provide a certain amount of protection for blood vessels, especially these big veins here called sinuses. A sinus is basically a very large venous structure that has very little supporting connective tissue contained within it because it's surrounded by other stronger connective tissues like the dura and the skull. So it's not reinforced much at all. It's just a great big blood vessel that makes it very easy for blood to drain into it because it's large and so the pressure is very low. And then the meninges also contain the cerebrospinal fluid. So it's something interesting to point out. Your body can make membranes that are capable of containing water. Water does not leak out of the meninges, but your cell membranes do not contain water or cannot contain water. So water does evaporate through the surface of your skin, but it doesn't leak out of the meninges. Moving forward then, so you do want to know those three terms there. The basic function of the meninges. Then you just want to be familiar with the description of each one. So the dura is the outer layer. It is the thickest and the strongest. It is made out of what I am assuming is dense regular connective tissue. I've never actually looked up what it's made out of, but it seems like it would be very similar to your joint capsules because they contain the synovial fluid, so they're also going to be structurally strong and waterproof, or at least highly water resistant. Then you have the arachnoid matter. That is all this web-like looking stuff here that they're calling the arachnoid trabeculae. So it kind of fills up this whole space. Some of it is attached to the inside of the dura, and some of it is attached to the outer surface of the pia. You just want to know where it is. And then the pia matter is attached to the surface of the neural tissue itself. And I like this diagram here because it shows that you have the arachnoid matter superficial to the blood vessels and the blood vessels superficial to the pia. I do believe in some places you can find blood vessels deep to the pia, but not so it would appear for your spinal cord. But there would have to be. I don't know, I have to look in to see if there are any blood vessels in here. I would almost imagine there would have to be, because it's a pretty thick piece of tissue to rely on diffusion. So that's it. That's your dura, arachnoid, and pia matter. Then you have the cerebrospinal fluid, which they contain. Basically, you want to know that they are very similar to plasma, the biggest difference being less protein. I'm not sure what the ion concentration differences are, but it's not hugely important for us. Presumably there would be some extra ions in there to make up for the fact that there's less protein. So you still have to have roughly the same osmolarity in every bodily fluid, just with different relative concentrations of protein and other solutes. So with fewer protein or less protein, you'd have to have more of another solute. What do we want to know? More importantly, what does cerebrospinal fluid do? It provides a certain amount of buoyancy for your central nervous system organs, which then protects them from blows and other trauma. And then it also helps nourish the brain. And because it also plays a role, this is relatively new stuff, in draining what I am going to call metabolic byproducts. Because that's really what they're talking about. Every once in a while, people will talk about toxins. Like they're things that you accidentally ingest and chemicals from companies. No, most of what your body has to deal with is stuff that your body makes. It's waste products, and you can't let it build up because it gets in the way of regular chemistry. So these metabolic byproducts have to be drained from the cranial cavity. And it does turn out that this happens quite a bit while you sleep. And there are some relatively newly discovered, they are not. There are lymphatic capillaries in your brain that are thought to help drain cerebrospinal fluid along with blood vessels to help, again, flush out metabolic byproducts produced by the brain that, if allowed to accumulate, would become toxic, but they're not foreign toxins the way most people think of toxins. I have a thing about that word toxins because people that are trying to separate you from your money will use it to get you to buy stuff that you don't need that is supposed to somehow magically cleanse your body of toxins. Try and eat healthy foods, drink enough water, and let your liver and your kidneys take care of your toxins. Moving right along, choroid plexus. This is where your cerebrospinal fluid comes from. So it produces cerebrospinal fluid. We did say that cerebrospinal fluid is produced by ependymal cells, so they are part of the choroid plexus. They enclose blood vessels, or capillaries, and then pump solutes from the bloodstream into the cerebrospinal fluid, and then water follows by osmosis. So the ependymal cells pick and choose what gets into the cerebrospinal fluid, and again, let the water flow by osmosis. You find them in your ventricles. This is supposed to be a peek into the lateral ventricle there, which would be all throughout here. The roof of the third ventricle there, and even a little bit in the fourth ventricle. Based on what I've seen in the sheep brain, I don't know how much choroid plexus is actually in the fourth ventricle, but you can definitely see it in the lateral ventricles. Third ventricle is hard to tell because if you cut right through the middle of it, you're going to disrupt the choroid plexus. But you can pull those blood vessels right out of the lateral ventricle when you do your dissection. And no, I do recall now you can find them in the third ventricle. They're there. So you can see the choroid plexus with the naked eye. That is where your cerebrospinal fluid comes from. Then what you want to know is how the cerebrospinal fluid flows throughout your dorsal body cavity, for lack of a better term. Right, so it's produced in your ventricles. If we start sort of farthest away from where it's going to end up in the lateral ventricle, it then flows through this foramen here, the interventricular foramen, sometimes called the foramen of Monroe. That leads into the third ventricle, which is connected to the fourth ventricle by the cerebral aqueduct. And then there are two apertures on either side of the cerebellum called lateral apertures. They are not shown in this image because this is a midline image, but they do show the median aperture. So there are three holes. There it is. Lateral aperture. Everything's blurry. That little hole right there. So cerebrospinal fluid leaks out of the brain. This is either through brain tissue or probably the pia mater. You're still enclosed within the dura mater here. It's just leaking out of the brain itself into the subdural space. Really, it's primarily in the subarachnoid space because the arachnoid tissue pretty much butts up right up against the dura. So it's in this space here, the subarachnoid space, where you find the cerebrospinal fluid outside of the brain and spinal cord. So it can go down and around the spinal cord and circulate around the brain, and then it is drained from your subdural space through what are called arachnoid villi. This book calls them arachnoid granulation villi. Why, I don't know. But it's a little protrusion of the arachnoid matter into or through the dura mater, and then into one of these big floppy veins called sinuses. Again, big low pressure blood vessels makes it very easy for fluid to leak into those spaces, right? And of course, this being your head, it can just flow by gravity down from your head to your heart. So you really just have a space for it, need to provide a space for it to go, and then it'll go from the sinuses into the carotid artery, subclavian, and then right into the heart. So that is your cerebrospinal fluid. We don't care about the sinuses or this thing here, the falx cerebri. So speaking then of blood and stuff, I should actually include the ependymal cells are not really part of the blood-brain barrier. So there are some blood vessels that are intimately associated with neural tissue. They're not just out in the subdural space separated from the nervous tissue by the pia mater. They get down into the tissues of the brain. Those blood vessels are covered by what we call the blood-brain barrier. And it really is just a series of structures that you find in capillaries in the brain that you may not find in other capillaries. It's the same basic capillary structure, just sealed up a little bit tighter. So you have a continuous endothelium, which just means there are no gaps in between the endothelial cells. All the cells are stitched together or stitched to each other with tight junctions. There are other capillaries in your body that allow for gaps so that you can just have bulk flow of all kinds of fluid out of the capillaries into the interstitial fluid, because it just makes it easier to deliver everything. But with your brain, your body is very particular about what it allows to come into contact with the brain. That's what the blood-brain barrier is about. So you have a continuous endothelial lining, a complete basement membrane, and again, that just means there are no gaps or pores, holes in the basement membrane. Some capillaries that are purposefully very leaky have pores in them. And then on top of that, the astrocytes also wrap themselves around. There really should be like a complete ring of astrocytes surrounding these blood vessels, and they just provide another barrier between the bloodstream and the neural tissue that surrounds it. So this is all there primarily to keep your neurons and your central nervous system free or separate from blood-borne substances, most specifically infection. So your brain or your body evolution has sort of rolled the dice here and not decided, because evolution doesn't decide things. But what seems to have worked out is that it is best to just make it really hard for an infection to get into the central nervous system. However, once an infection gets in there, because of that blood-brain barrier, that are just structures that can't differentiate between a virus and a macrophage or a bacteria versus a neutrophil, it keeps the cells and proteins of your immune system out the same way it would keep bacterial cells and viral particles out of your brain. So once infection does get deep to your meninges, once you have meningitis, bacterial or viral, it can be very difficult to treat because your immune system doesn't get in there very easily. But it must be that this is the better way to do it, to just make it really hard for the infection to get in there in the first place. That keeps your central nervous system safer than granting your immune system access to it. Because any portal of entry that your immune system could use to get into your brain, a virus or bacteria could use as well. And you don't really want your immune system going full force into your central nervous system, because your immune system releases a lot of inflammatory chemicals while it's trying to fight infections. You experience side effects of your own immune system when you're sick, and you don't want your brain experiencing those side effects or having massive inflammation within your meninges. All right. Now we are on to the last two things, and I should point out I made some changes to the objectives if you are listening to this podcast in the fall of 2020. These are new changes. If you go to the link, you'll find them in the link. So that file, which is in Dropbox, has been updated, just so you know. Whoops, let's go back here. So the blood-brain barrier I added, and then I took out the spinal cord because we don't cover that in lecture anymore. There's a bunch of stuff that we've just cut out, and I've simplified to a certain extent the reflexes by cutting one of the reflexes out. So the one reflex that you do want to know in detail is the stretch reflex. This is the patellar stretch reflex. Any of the reflexes for any muscle where the physician or clinician is banging on you with a hammer is a stretch reflex. Just to sort of give you the description of why it happens the way it happens, what you're simulating here is an involuntary stretching of the muscle. So in this case, your quadricep is getting longer, and it's not because your hamstring is getting shorter. This is what sets off the reflex. So your body basically thinks that your leg is buckling and you didn't make it happen. So the way to prevent that from happening is to contract your quadriceps, straighten out your knee so you don't fall down, because from your body's perspective, your knees are bending, you're falling, and it is not a planned fall. So the reflex is to extend the leg so you don't fall down. So what you want to know is what is going to happen to each neuron and at each synapse within the reflex. So we start here with the stretch receptor, which is called the Golgi tendon organ, but we don't necessarily need to know that, right? This yellow sensory receptor is going to get excited. It will ultimately make your brain aware of it, but the reflex that we're going over does not involve the brain. So this happens before your brain even really has a chance to respond to it. So sensory neuron gets excited. It is going to dump excitatory neurotransmitter onto a motor neuron and an interneuron. So those little green pluses mean excitatory neurotransmitter. This purple interneuron releases an inhibitory neurotransmitter. So what's going to happen is that the motor neuron that controls your quadriceps, which you want to contract to straighten out your leg, is going to be stimulated. That is then in turn going to stimulate your quadriceps, and they are going to contract. At the same time, you want to ensure that your hamstrings stay relaxed. And you need to consider that this reflex isn't always happening when you're sitting in the doctor's office and you know it's coming. It is designed such that it can override other motor instructions that might be coming down the spinal cord from the brain at any point in time. So if you're in the middle of a motion and you start falling, like the stretch reflex gets activated in a real life action situation, the reflex circuit is going to override any conscious motor instructions coming down to your legs. So this interneuron is going to dump inhibitory neurotransmitter onto the motor neuron that controls your hamstring. What that means is that Nothing happens at your hamstring. So if you recall when we did muscles, your motor neurons release acetylcholine. That's it. So the signal from neurons to muscles is a contract signal. There is no relax signal. In the absence of a contract signal, the muscle just relaxes. So in order to ensure that nothing happens here at number 8, you have to have inhibition here at number 7, because there might be a motor neuron from the cortex coming down here that's going to dump some stimulatory neurotransmitter onto this motor neuron here, because maybe you were trying to do something that involved contracting the hamstring, but it went wrong, and now this stretch reflex gets activated. This purple interneuron is going to override any stimulatory input from other neural circuits and make sure that nothing happens here, so that you only get contraction here and this muscle stays relaxed. So you want to know, well, everything that's written here, right? Yellow motor neuron gets excited. Green motor neuron gets excited. Interneuron gets excited. Green motor neuron stimulates contraction. Purple motor neuron inhibits red motor neuron so that nothing happens at the hamstring. So all of that, everything that's next to the numbers, be aware of that. That is the stretch reflex. That is the only one that you need to know in detail. And then the opposite of that is the flexor reflex. And this is done to pull a limb back from stimulus. And you don't need to know any of the wiring here, because we're not even going to worry about it. There are different variations on the flexor reflex anyway, some that involve multiple interneurons. This one just is a simple 3-neuron reflex arc. But just like this reflex arc here, there probably is in actuality a inhibitory interneuron that is making sure that the antagonist of this effector stays relaxed. So it's probably very similar to this in the wiring, just with a different relationship to the muscle. So it's the same wiring set up to cause flexion of the limb as opposed to extension of the limb. So a flexor reflex of the knee would look like stimulating the hamstring and making sure nothing happens to the quadricep. So this green motor neuron would be inhibited by an interneuron. More than you need to know. This is what you need to know right there. Pull the limb back from the stimulus. Crossed extensor reflex, we don't do. But I'll tell you what it is. It's a flexor reflex on one side when you are pulling away from stimulus, with an extensor reflex on the opposite side. So if you quickly pull back your left arm, your right arm will go out to balance you, right? If somebody pulls on your left arm, you're probably going to stick your right arm out reflexively, because that's how you maintain balance. Or if you step on a tack with your left foot and you pull your left foot up, you better darn sure put your right foot down or you're going to fall. That's the crossed extensor reflex for your legs. That's it, 23 minutes. That's the end of this chapter for this week. Thanks for tuning in.